Thank you, Martha. And I'd like to thank the organizers for the opportunity to speak with you this morning about these new agents that have been developed to treat systemic amyloidosis. These are my disclosures. I'll follow this simple outline this morning, covering the highlights of these classes of agents and some of the clinical benefits. I'll also present preclinical data on novel agents that may be part of future therapies. As everyone knows, the two most common types of systemic amyloidosis are ATTR and AL. ATTR can be hereditary or wild type. ATTR-M, the hereditary form, is caused by one of over 100 mutations in the TTR gene, resulting in more rapid dissociation of the tetramer into 4 monomers. In AL, as in ATTR, there are cellular and extracellular phases of protein production and disease pathophysiology. RNA interference deals with the production phase, and monoclonal antibodies can deal with the cells as well as with several extracellular phases. ATTR, as you all know, is a disease in the hereditary form that has a variable mix of cardiac and neurologic symptoms. In both instances, it is a progressive disorder and can cause significant disability, wasting, intractable autonomic neuropathy, and end-stage heart disease in less than a decade after the onset of notable symptoms. At the same time, even in areas such as the US, where the African American population has a prevalence of 3.4% of isoleucine 122, and in areas like Portugal, Japan, Scandinavia, and County Donegal in Ireland, where the prevalence ranges from 0.1% to 1%, the penetrance is not well defined. ATTR wild type is a clinically similar but somewhat different disease. The connective tissue involvement with biceps tendon rupture and spinal stenosis, and we have over 300 cases, which we'll be presenting in Tarragona, of patients with spinal stenosis whose ligamentum flavums were stained with amyloid. The percentage of amyloid is around 20%. It's not 100%. If it were 100%, the country would be spending $20 billion on surgery for spinal amyloidosis, and corporate interests would peak notably. In any case, it is likely the cause of HFpEF, as Martha said, in 10 to 15% of patients with HFpEF over the age of 75, equally distributed among men and women. Most of TTR is produced by hepatocytes, the cells that are shown on the slide. And normal TTR is a chaperone protein for vitamin A and thyroid hormone. This schema depicts the dissociation of the tetramer into monomers, and the monomers are the vector of ATTR amyloid. It also shows the misfolding, the oligomerization, and the beta fibrillation stages. The cause of AL is misfolded free light chains produced by clonal plasma cells. Normally, within plasma cells, light chains are produced in slight excess of heavy chains because unpaired heavy chains are not secretory competent, and within the plasma cell can misfold and trigger an unfolded protein response that can be terminal. Light chains, in contrast, are secretory competent, and in normal persons circulate at levels of up to 25 milligrams per liter. A functional physiologic role for circulating normal light chains has never been defined to this time. In AL, there is obviously lambda predominance, and as Dr. Schoenland showed, a predominance of the 11-14 translocation, resulting in loss of the heavy chain and hence light chain disease, and also in some cases loss of a microRNA cluster that's probably important. This results in loss of the, and although the entire light chain, the variable, the joining and constant region can be involved in amyloid beta fibrillation. The small number of light chains that are more prevalent in AL than in myeloma demonstrate a pattern of organ tropism that has been described in other plasma cell diseases. RNAi aims to stop translation, stop protein production. In ATTR, the culprit protein is obviously transthyretin, and in theory, in light chain disease, it could be the free light chain genes. These agents are highly specific, and the activities, the different half-lives, the rates of decay of messenger RNA within cells are extraordinarily complex and important features of cellular metabolism. RNAi takes advantage of two of these major degradative processes. For antisense oligos, say 20 nucleotide stretches of the oligo include flanking portions of RNA nucleotides and a mid portion of a DNA stretch of nucleotides. This agent binds to messenger RNA, creating a hybrid. And that activates one of the major degradative processes intracellularly, RNase H1 degradation of the hybrid. This is sort of like the state police, maintaining homeostasis within the cytoplasm with respect to translation. I compare the formation of the RNA-induced silencing complex to the FBI. So siRNA is a double-stranded RNA molecule that separates in the cytoplasm. The sense strand binds to an appropriate risk and triggers degradation of the message that is its target. However, there are many challenges for RNA interference. Getting from outside the body into the circulation and to the target tissue is particularly challenging. There are nucleases and proteins in the circulation that rapidly degrade circulating nucleic acids. Many vehicles and many modifications have been used over the years to try and solve this problem, and it turns out that lipid encapsulation is likely the most effective way we currently know to solve the problem of delivery. At the same time, encapsulated RNAi agents in lipids are more likely to go to the liver, and hence the focus on liver diseases such as TTR amyloid and porphyria. It's very important to understand that the next phase, once the lipid has entered the cell, is also very complex and requires trafficking within the cell to the appropriate degradative sites. And that's an issue that's been worked out empirically by the commercial entities. Inotersen is an ASO, and it's approved for the treatment of hereditary amyloid with peripheral neuropathy. It was developed and approved on the basis of a phase 3 randomized trial. A validated peripheral neuropathy instrument, the NIS plus 7, was used to define the primary endpoint. Quality of life instruments were used to define the secondary endpoints. Prealbumin, the tetramer of TTR, was also measured, and cardiac measurements such as wall thickness were used as well as correlative endpoints. So interestingly, the patients who received inotersen, over half of them had reductions in prealbumin, so they had reductions in TTR. The secondary cardiac endpoints, by and large, were very excitingly met. And the evidence for impairment of progression, reduction in progression of cardiac amyloid, the evidence was quite strong. However, there was a 22% withdrawal rate in the inotersen arm, an 8% in the placebo arm, mostly due to adverse events and progression of disease. There was a 3% of incidence of glomerulonephritis in the inotersen arm, and also more frequent thrombocytopenia of all grades. There were 5 deaths in the study, all in the inotersen arm, and one of the deaths was due to intracerebral hemorrhage in a patient with grade 4 thrombocytopenia. The drug is approved. It's given subcutaneously on a weekly basis. Patisiran, a second RNAi agent that is an siRNA agent. It's given every 3 weeks IV as a stable nucleic acid encapsulated lipid particle. The cargo is the siRNA against TTR, the TTR message. In the phase 3 trial that led to its approval, the same instrument to score peripheral neuropathy was used, the NIS+7, and quality of life instruments were also used for secondary endpoints. The withdrawal rate was 38% in the placebo arm and 7% in the patisiran arm. There were infusion reactions that were generally mild in 20% of patients in the patisiran arm. The graphs that I show here are very interesting. The graph on the left shows that the TTR level measured as prealbumin is actually significantly lower in those who got patisiran. However, not all of them had improvements in NIS+7 scores. That means that you can't use the prealbumin level as a marker of response to patisiran. You have to use the patient's clinical status. Cardiac improvements also occurred in the subset of patients with cardiac disease. This agent is approved and, as I said, is given every 3 weeks intravenously. This patient came to me late in 2017, quite a history of atrial fibrillation. He developed orthostatic hypotension that dramatically affected his golf game and had a family history that was interesting. He was of Irish descent. His grandfather died of amyloidosis in County Donegal. We diagnosed the ALA60 very quickly. Fortunately, there was a trial available for him, the PRX004 trial, which I'll mention shortly. He enrolled on the study. He tolerated the agent at the lowest dose level. Obviously, he was first patient in. And then went on to receive patisiran once it was approved. His prealbumin level went down immediately on patisiran and it stayed low. His orthostasis essentially resolved in about 6 to 8 months. His atrial fibrillation remained a problem. He had an ablation and a dual chamber pacer placed, which in one instance in the past 6 months has turned out to be life-saving. He also had really massive diarrhea when we diagnosed him. That has largely resolved now at 18 months, although he still has bad days. He'll have a bad day every 6th or 7th day. The graph on the bottom of the slide shows what has happened to his vitamin A level. Obviously, it's gone down with the suppression of circulating prealbumin. So he's on supplemental vitamin A, which is a necessity. So with the success of siRNA technology, we decided to try and knock down light chain production. We have a bioengineering collaborator who's been working with us. We've designed siRNAs for the constant region of the kappa and lambda light chains. We can deliver the siRNA successfully in vitro. The top left-hand slide shows a labeled Sci5.5 siRNA that has been delivered into plasma cells. We can successfully knock down intracellular light chain in patient cells, the middle slides on the top. And the far right slides show that we can knock down production in vitro with siRNA directed at the constant region of the light chains. So we developed xenografts in NSG mice using 2 myeloma cell lines. The xenografts engraft very quickly, very short latency period, 12-day latency period, which is very important as you're trying to develop effective technologies. The cells are intraperitoneally injected. What you see in the lower left is an image of the mice at 12 days and an image of a liver section from one of the mice showing the CD138 positive human cells. The image in the middle on the bottom is an image of the lipidoid nanoparticles, which are injected beginning on day 5. And what we see is that by day 8, in these rapidly engrafting human myeloma cells that are highly productive of light chains, that we can mute the production of light chains in a 3-day period. One of the interesting things about these reporter cell lines is that we can correlate light chain production with the bioluminescent signal and with soluble BCMA, which I'll say a little more about in a second. So we've got numerous ways of assessing tumor burden. Which brings me to monoclonal antibodies. The other class of agent that has great relevance to systemic amyloidosis. From suppression, therefore, we go to scavenging. I'm going to talk a little bit about monomer scavenging, and I'm going to talk a bit about fibril targeting. But what's interesting about monoclonal antibodies is how sophisticated the universe has become. Monoclonals can target cells, they can target proteins, and now we know they can target infectious agents. The success of monoclonal antibodies in treating Ebola is really astounding, truly astounding. One thing that we know is that in the setting of RNAi, the lipids, the coating, the design of the agents within the lipid capsules, all of those elements give a variability which allows for the development of intellectual property, which allows companies to become interested in things because they can patent unique formulations. The same applies to monoclonal antibodies, as this sketch suggests. There are ways to mix and match elements of immunoglobulins that can be uniquely used and may be of great benefit. Etanercept comes to mind. Etanercept puts the receptor for a cytokine, 2 of the receptors, on an FC portion of an immunoglobulin. And it's FDA approved and it works. And the technology itself, evolving from hybridomas to phage display libraries, to humanized mice. It's created a global commercial effort that is really, really extraordinarily beneficial for humankind. But there are still challenges, and amyloid obviously provides major challenges for the application of monoclonal antibodies. XO4 is an anti-TTR monomer, and it's in clinical development. We had the first 2 patients on the study. The trial has moved along. It's a phase 1 study. Radhika is available to you if you're interested in where it is and whether there are slots open. But one of the unique aspects of hereditary TTR, as this gel from Lorraine Connor's lab at BMC shows, is that you can see the lower band. That's the wild type monomer. It's migrated to this isoelectric neutral point in the gel, and all of those bands are at the same level. The mutant form of the monomer migrates to different levels. And this is probably why there's no lab test for the mutant TTR monomer available to us. The suppressors have prealbumin as a readout for efficacy, but agents directed at the TTR mutant monomer do not have an assay available to them. We're really hopeful that as a result of this trial and the work of Prothena in this area, such an assay may be developed. And of course, there's KAL101, the anti-amyloid antibody that's still standing. It's very promising, and I do think that excitement exists at the fact that Alexion has confirmed they will be conducting a clinical trial. Hopefully we'll be starting next year in patients with advanced AL amyloid. It'll be an advanced stage trial, we hope. As you know, this antibody was first produced in Dr. Solomon's lab and was tested clinically in a phase 1 trial by Dr. Lynch at Columbia. The trial that Alexion plans will accrue a couple of hundred patients, and we certainly hope that it gets underway soon because patients need to have this question answered. As you know, the history of this antibody dates back a decade. The image on the left of this slide was on the cover of Blood in 2010, and it demonstrates that the antibody binds to amyloid in organs. The images in the middle show that it can activate resorption of amyloidomas, these surrogate tumors in mice. And the image on the right is from the clinical trial, showing that use of the agent may have a cardiac benefit in improving global longitudinal strain. And we move now to a look back at the ride Prothena gave us with D1, and it was quite a ride. D1, as everyone knows, is no longer in clinical development. We hope that the biobank and the control arm data from the D1 trials will be made publicly available and that productive efforts can be made to learn from those data. The preclinical data for D1 were robust, but not as robust as the KL101 preliminary data. Wagner Zago for Prothena showed that D1 did bind to soluble aggregates in vitro and had a lovely motion picture showing that with a macrophage cell line, opsonized amyloid aggregates could be phagocytized. Very, very exciting little movie. In the phase 3 VITAL trial for newly diagnosed AL patients, a trial that was halted because of futility, there was the hint of a benefit based on the hazard ratio. And when subset analysis was done, and this is published now, what we saw in the Mayo stage 4 patients, these are patients with stage 3 cardiac and very elevated light chains, was an apparent survival benefit. Those who got CyBorD without D1 had the typical 6-month median survival. Those who got CyBorD with D1 had improved survival. Very suggestive data. Dr. Gertz has a poster, number 3166, describing this data, and I do think it's well worth looking at. When I saw this data initially over a year ago, I said, well, D1 must be sopping up the aggregates in these patients because they have so many light chains. That is a total speculation. Some might call it an hallucination, but that was my explanation for it. When we looked at the code and broke the code and identified those who'd gotten D1, we learned that we had treated a small number of patients with both D1 and daratumumab. And these data are being published. They'll be out within days to weeks. And what we knew at the time was that daratumumab was very active in myeloma. It had been approved in November of 2015. We didn't know whether or not it would be active in AL, but it made sense to use it in patients who were very sick and getting sicker. The Andromeda data that Dr. Zonder referred to is shown on the left in the bar graph. And the data on this small cohort of patients who got dual monoclonal antibody therapy is shown in the other sketches. Of particular interest to us is a single case, the free light chain response, the cardiac response in the lower graphs. This was a patient who started with an NT-pro just under 8,500, a free lambda just under 1,000. Both rose, as you can see, in the early phases of treatment. And then with the administration of daratumumab, both came down relatively concurrently, which is a very unusual event in amyloid. Usually the cardiac response is out of phase to the hematologic response. Now, we believe that these data are important. We believe that dara may enhance anti-amyloid antibody therapy because of its impact on T-cell subsets. and also would urge interested pharmaceutical companies to consider dual antibody therapy for patients with AL, because these were very sick patients, and yet they tolerated 2 monoclonals. And so we come to future therapies. Dr. Zonder didn't mention anti-BCMA immunotherapies, but I think they're going to be extraordinarily important for AL patients. He did mention clinical trials, and it is my hope that Belmaf will be available in a clinical trial next year, thanks to the efforts of a small group of investigators and collaborators at GSK. One of the interesting aspects of the murine models I mentioned was that BCMA correlates with flux and light chains, and that's what that graph shows, the correlation between BCMA and kappa light chains in the mouse model. The image on the right is Belmaf, which is an antibody-drug conjugate. On Monday evening, Dr. Gadar from our group will present our data on BCMA and AL. BCMA is found on the membrane of AL plasma cells. Soluble BCMA can be measured in the serum. You can modulate BCMA on the surface of AL plasma cells with gamma secretase inhibition. And when you do that, the amount of soluble BCMA in the supernatant in vitro goes down. So we're very hopeful that this agent will go through the clinical development process and be available to us within 2 years. And you can envision a treatment plan that might include dual monoclonal antibody therapy up front or in patients with refractory disease in the future. And finally, what about these circulating culprit light chains? There's no maintenance therapy for AL. In another poster that Dr. Gadar will present on Monday evening, what we show is that the current response criteria for AL are probably outmoded and in need of revision. Particularly what we show in a cohort of 133 patients who achieved VGPR between 2006 and 2017 is that long-term survival in 42 patients who had involved free light chains below 10 milligrams per liter for any durable period, a month, 2 months, 3 months, the median was about 3.5 months, had 95% survival at 10 years. And not all of them had achieved CR. About 20% of them were VGPR patients. The rest of the patients had a median survival of 10 years. So when you look at this disease, you have to keep in mind, as Dr. Zonder suggested, that in patients, for example, with renal failure, if the involved light chain isotype is elevated, there's probably some culprit light chain there that's causing progression of disease. Don't forget, when we reduce the light chain culprit, we slow the progression of disease. But that doesn't mean that we necessarily stop it. So I do believe that the monoclonal antibody that's being developed may provide an effective agent maintenance. We've done surface plasmon resonance testing, which you see here. We have shown in vivo activity in our mouse model, which we'll present in Tarragona in March. We're humanizing the monoclonal, and we're testing it in vitro against patient samples. The goal would be to use it not up front, but in maintenance to markedly reduce the level of the culprit light chain isotype. So this satellite symposium documents the great interest in amyloidosis, as Dr. Grogan has indicated. There are multiple new classes of agents. There are suppressors, stabilizers, scavengers, RNA interference agents, monoclonal antibodies, and small molecules like AG10, which will likely be very important. And all of those classes attest to the golden age that we're now participating in. I might call it an apple-green birefringent age in pharma, but it's certainly a golden age for patients and for the physicians who struggle with this disease for decades. And as Dr. Grogan suggested, this is just the beginning. The RNA agents that are being developed in second generation are different. They don't have to be dosed as frequently. And they're being developed, we hope, for other types of amyloid. Monoclonal antibodies are being tested against other cell surface proteins and will be, I suspect, developed for other culprit amyloid proteins. The lab-based technologies are important as well. The use of next-generation sequencing for organs that are affected by amyloid, I think, should be front and center of things that we're interested in, because we have to come to an understanding of the processes, as Dr. Zonder suggested. What's the effect of tissue displacement by amyloid? Is there a potential ischemia going on in small vessels? What actually triggers the fibrosis that we see in the organs? And we need to have much better understanding of the pathogenesis of organ disease. Robust patient advocacy organizations are extremely important. The Amyloidosis Research Consortium held a patient forum in Washington, D.C. early last month, showing the interest and the willingness of the regulatory agency to engage in the conversation with pharma. Very important benchmark. And finally, I do have to thank our cardiology colleagues, not only Dr. Grogan, but Dr. Dorbala, Dr. Falk, Dr. Ruberg, and Dr. Maurer, for all of their efforts in cardiology. It's inspirational to see the efforts that are being applied to control that wild west out there of PYP scanning, as Dr. Grogan suggested, for patients with HFpEF. Finally, thank you for listening to me this morning. And please join me in committing to earlier diagnosis of these patients and novel clinical research. And I'd like to thank those who've supported the work I described and the organizers again for letting me speak this morning. Thank you.